This video will demonstrate how to perform SDS page for protein characterization. SDS page stands for sodium disulfide polyacrylamide gel electrophoresis. Here are the equipment you will need. First, insert a short plate and a 1mm spacer plate into the casting frame. Make sure the feet of the casting frame are on the bottom. Make sure the plates are also level at the bottom. Then tighten the clamp to secure the position of the plate. Next, install the casting frame with the plate onto the casting stand and ensure the setup is tight and secure. Check the seal of your setup by pipetting DD water halfway up the plates. Observe if any DD water leaks. If there are no leaks, then remove the DD water either using clean wipe or filter paper until it is almost dry. When the gel casting stand is set up, prepare the 12% resolving gel solution in a 50mm Falcon tube. Mix 3.3mm of DD water, 4.0mm of 30% acrylamide solution. Liquid acrylamide is neurotoxic and should be handled with caution. 2.5 ml of flour stock and 100 microliters of 10% SDS together. Right before you are ready to pour the gel, add 10 microliters of 10% ABS and 4 microliters of TMAT. TMAT catalyzes the polymerization of acrylamide. If TMAT is added way too early, the gel will set before it is ready to be pipetted in between the plates. Mix gently, then immediately pipette resolving gel solution in between the plate with the glass pipette until the resolving gel reaches just below the grain line or approximately 1 cm from the top of the short plate. Pipette 200 microliters of isopropanol on the resolving gel and observe the thin layer on the gel. While waiting for the resolving gel to polymerize, prepare the stacking gel solution in another 50mm Falcon tube. Mix 1.35 ml DD water, 0.335 ml acrylamide stock, 0.25 ml upper stock, and 20 microliters of 10% SDS. Once the resolving gel has formed, which takes roughly 30 minutes, drain the isopropanol. Gently rinse the top of the gel with DD water and dry with clean wipes. Right before you are ready to pour the stacking gel, add 20 microliters of 10% ABS and 2.0 microliters of TMAT into the stacking gel solution and mix well. Immediately pipette about 1 ml of the stacking gel solution on top of the resolving gel in between the plates. Fill to the top, then insert the comb into the solution. Wait for the gel to polymerize. After the gel is formed, unclip the casting frame with the gel from the casting stand. Then carefully remove the glass plate from the casting frame. Wrap the plates carefully in wet paper towel and saran wrap, then store in the cold room until next week. To prepare samples for SDS page, label the microfiche tube corresponding to the samples. Pipette 40 microliters of the fish extract into the microfiche tube and add 40 microliters of the sample buffer. Heat the tube at 100 degrees Celsius for 3 minutes, then pulse centrifuge for 5 seconds. Remove the gel from the saran wrap and paper towel. Tightly secure the gel with the short plate facing inward onto the running module. Each running module can hold 2 gels. If only one gel is being run, need to use the buffer dam in place of the second gel. Do not directly label the plate with tape because this will prevent the gel from running properly. Instead, plate can be directly labeled with a permanent marker. Each tank can accommodate 2 running modules, which will be 4 gels altogether. Insert the running module into the tank. Make sure the module properly insert into the slot in the tank. At this point, check to see if the lid fits appropriately. Pour the 10 times diluted running buffer solution into the running module between the 2 plates until well on the 2 plates are fully submerged. Check to make sure the running modules aren't leaking any buffer solution into the tank. The tank only needs to be filled halfway with the buffer solution if running 2 gels. Now, ready to load the samples. Remove the comb so we can see each well clearly. And we can also install the loading guard on the gel to make it easier to load the sample properly into each lane. Carefully load 10 microliters of sample solution into each well. The pipette tip should be inserted into the well in between the plates, deep enough to prevent overflow of the sample to the neighboring lanes. Slowly release the sample solution into the lane, and when complete, remove the pipette tip, change the tip, then load another sample. For the molecular weight marker, we also need to load 10 microliters into the well, and each will be loaded in one of the center lanes for easy comparison. When all the samples are loaded, close the lid on the tank and insert the electrode properly into the power supply. Red wire into the red slot and black into the black slot. Set the power to 20 mA per plate and run for 1 hour. The power can also be set as voltage. The voltage used does not depend on the number of plates, so a consistent voltage can be used to run any number of plates. For example, a consistent 120 volts can be used. Should initially observe the sample or standard solution to be condensed into a thin band through the stacking gel. Then the band will gradually migrate down the resolving gel. When the blue dyes have reached close to the bottom of the gel, stop the run. Unplug the power supply, then remove the running module from the tank and drain the running buffer. Remove the glass plate carefully from the running module, then carefully use the gel releaser to remove the gel from the glass plate. You can trim off the stacking gel carefully before proceeding to staining. Transfer a gel into a square petri dish, and then pour the Comazi blue staining solution into the petri dish, submerging the gel fully. Place the square petri dish in a metal tray to avoid spillage in the incubator, then place in a 50 degrees Celsius incubator to stain for 30 minutes. Remove the gel with a tweezer and make sure you can observe clear band on the gel before discarding the staining solution and replacing it with the destaining solution. When destaining, add a pieces of Kim wipe or absorbent paper towel to help absorb the dye. Destain at 50 degrees Celsius for 30 minutes with periodic swirling to facilitate the destaining process. When the destaining is complete, store the gel in the water until you can scan and analyze it with the densitometer. Properly dispose of any liquid acrylamide containing solution in the acrylamide waste container. Polymerized gel can be disposed directly in the garbage, while destaining and destaining solution can be disposed into the non-halogenated waste container.